Hi, my name is Tom Goodwin, and today I'll be presenting about bioinspired liquid-infused membranes for ultrafiltration. I've been doing this research with Professor Jessica Schiffman and graduate student Rushab Shah. So, we all know that water is essential to human health. But unfortunately, 1 in 3 people lack access to clean drinking water in the world, and there are over 2,000 deaths every day from unsanitary water. Now, a popular method for water purification is ultrafiltration, which involves membranes with a pore size of 5 to 100 nanometers. This small pore size removes pathogens and unwanted particles very well and is ultimately very helpful for stopping waterborne diseases. Ultrafiltration is also very cost effective and energy efficient, so it's one of the top methods of filtration right now. Now, one of the biggest problems with membrane filtration in general is fouling, which is the accumulation or adhesion of particles on the surface or in the pores of a membrane. So we can see in this diagram on the left that the particles block the pores of a membrane, which reduces the flux. Now, reduced flux leads to reduced filtration efficiency, and eventually membranes will become useless from fouling and can even be damaged because of the pressure differences caused. Some examples of foulants are pathogens like bacteria and viruses, macromolecules like proteins, and even dust particles. Now, a solution to fouling is liquid-infused membranes, which are inspired by the pitcher plant. So we can see in this picture on the left that ants are able to crawl all over the dry surface of the pitcher plant. But when the pitcher plant secretes a lubricant to its surface, the ants are no longer able to crawl on the surface and slip. We want to try and mimic this and basically treat ants as foulants in our case. And by doing this, we will add a stable liquid to our rough porous surface to create a smooth, defect-free surface that will repel foulants. Now, we use polyvinylidene difluoride membranes, or PVDF membranes, and for the lubricant, we use Krytox 103 and 107 perfluoropolyether oils. Now, PVDF membranes are good because they're very inert, like chemically inert, and they also are structurally very stable. The Krytox oils are good as the lubricant because they have low surface tensions, so they'll have a lot of potential of lubricating the membrane, and they're also immiscible with water and hydrocarbon oils. Now, to fabricate our liquid-infused membranes, we pipette 10 microliters of lubricant per square centimeter onto the membrane. We then let the membrane rest horizontally for half an hour so the lubricant can fully saturate it. And then finally, we'll rest the membrane vertically for 1 hour, and this will allow all the excess lubricant to flow off the membrane. Now, how do we test if we've actually fabricated liquid-infused membranes? Well, we used the crystal violet stain test. So this test involves soaking our samples in an aqueous crystal violet solution for 15 minutes on an agitated shaker plate. And this basically will cause any exposed membrane to die because this crystal violet stain is hydrophilic and will stain the membrane, whereas the hydrophobic oil prevents the membrane from being stained and does not interact with the dye at all. So we can see on the left of this picture that the bare membrane is completely dyed, which is what we expect. But on the right, we see the 2 Krytox-infused membranes do not dye at all, basically, because that oil is preventing the hydrophilic stain from contacting the surface of the membrane. Now that we know that we've actually successfully created liquid-infused membranes, we will perform flux experiments. Now, we're very interested in flux properties because they're not well studied, but they're very relevant for filtration applications. So to do this, we use a dead-end filtration unit, which involves pumping water through a membrane using pressurized nitrogen, which we can see in our picture on the right. And then we collect this water in a permeate bottle. So to perform pure water permeance tests, we pressurize deionized water at 1, 2, 3, and 4 bar and allow this water to flow through our membrane or liquid infused membranes at each of the pressures said before for 15 minutes. And then during those 15 minutes, during each minute, we measure the mass of the water collected in the permeate bottle. And using this mass measurement, we can actually calculate the flux that's passing through the membrane. And then from the flux, we can calculate the permeance, which is a measure of how permeable the membrane is. So now that we know how to do these experiments, we can perform controlled experiments varying a single parameter of each liquid infused membrane. So for this experiment, we varied the pore size. So every other parameter was constant. It was a hydrophobic base membrane, a PVDF membrane actually, and we use Krytox oil as the lubricant. Now, we can see in the data on the left that the 0.2 micrometer pore size membranes had roughly constant permeance for each pressure, but the 0.45 micrometer pore size membranes had increasing permeance with increasing pressure, which means that the Krytox 103 does not behave the same in the pores for each of the different pore size membranes. Another experiment we performed was varying the surface chemistry of the base membrane. So like I said before, we basically kept all of the characteristics of our liquid infused membranes the same, except those base membranes, one has a hydrophobic surface and one has a hydrophilic surface. So we can see in the data on the right that the hydrophobic base membrane limbs had roughly constant flux or constant permeance. Whereas the hydrophilic also had very similar permeance, but it varied a little bit more. But these error bars are all overlapping, so we can't say that there's a significant difference between permeance values at each pressure. We also can't say anything, we can't say that there's a significant difference between the hydrophobic and hydrophilic permeance at each pressure. So what can we conclude from all this data overall? Well, to recap, we know that we successfully fabricated stable liquid infused membranes. We know that the lubricant behavior changes with pore size. And we can even hypothesize from this data that smaller pore sizes lock in the lubricant better, which may be why we saw more constant permeance. Whereas larger pores may have the lubricant flushed more, which may be why the permeance was increasing with higher pressure. And then we know that the lubricant behavior appears to be a bit different for the surface chemistries, but it's not statistically significant enough to conclude anything. And we need to perform more tests to actually determine if hydrophobic or hydrophilic base membranes are important. Finally, we know that this project is far from done, but we want to perform a lot more experiments. So one of them is a molecular weight cutoff test, which basically involves dissolving different molecular weight polymers in solution and then passing that solution through our liquid infused membrane and seeing what the maximum size polymer is that can pass through. And this will basically tell us what the pore size of our liquid infused membrane is. And then we also would like to perform total organic carbon readings on the permeate of our liquid infused membranes. And this will basically tell us how much of the lubricant is coming off, if any at all. This is all the data I have to present today, and I'm looking forward to presenting more data in the future. Thank you for watching my presentation.